Good afternoon, I'm Pat Bradshaw. As Dr. Russell and Hugh have both told you all, that I'm program manager for Bioenergy and it's going away, so I'm not sure if that's reflecting on me or whether that's what that is.
Yeah.
Thanks, Hugh. And I suppose next I'm going to be charged for a BRI, so I'll end up owing you money.
It helps when you put it on your tie.
Better? Better?
Higher. They want it higher. There you go.
All right. How's that? Good. So I don't need to tell you how important energy is to the Air Force. My program is not in producing major groups of or information, a lot of energy. It's just to find out the mechanisms that go into producing bioenergy. As Hugh had said, I get to report on everything Walt set up. And Walt was here in this program for 7 years. He did a great job. He set up a great team. He had researchers that are doing some just wonderful stuff, and I get the opportunity to report on what they're doing. We'll go into where the proposal or the program is going to be going. But what is bioenergy? Well, it's an area that's characterized by modeling and explaining the structural and metabolic functions and gene regulatory mechanisms associated with capturing energy. The areas that I'm going to talk about is biosolar hydrogen, algal oil, and biofuel cells. I'm not going to talk much about photosynthesis. That's about 80% of the portfolio. The other 20% is the area that we're going into. It's a fairly new portion of the portfolio where they're trying to focus on understanding the genetic molecules and biochemical mechanisms, as well as tracing the neural pathways involved in stimulating cells, tissues, and organs. So when given the question of can you develop a biofuel produced from carbon dioxide, water, and sunlight, the obvious answer came in, yes, we can do that through algae. Algae's been studied for many years. Way back in the 70s, they were looking, during the first energy crisis, they were looking at producing oil. When the prices went down, they stopped the funding for that area. When asked the question, can you develop a portable hydrogen fuel generated from water, cellulose? Yep, they can do that. The engineers or the researchers came about and did a great job on that also. I'll be talking about both of those. And for compact power and ambient biomass, the biofuel cell work is astounding. So Walt set up a tremendous team. He did a great job. He was a great coach. And each one of the researchers will tell you that they were surprised that when he wanted people to work together, he would direct them to work with this team and them to work with that team. And they were very happy with what he actually set up. So, last level or area that I get to talk about is photomagnetic electrobiostimulation. It's a new emphasis area, and the laboratory is out in front working in that. Here's where we're going. We're going to decrease in biosolar hydrogen, as Dr. Russell told you. We're going to decrease in algal oil. We're going to increase in synthetic biology. We're going to stay about the same for enzymatic fuel cell and microbial fuel cells and artificial photosynthesis. So we'll keep kind of a tech watch in that area until maybe some of them run out, or until we decide whether we want to get completely out of them or not. Areas will increase photoelectromagnetic stimulation, biomarkers, physiological responses in toxicology, and synthetic biology. When we look at nature, we try to characterize mechanisms for the basic science portion. We call that like the first generation of science. As we start to understand it a little bit better, we try to optimize or understand how to optimize the system. We get into more metabolic and protein characterization. That's still on the biology discipline. A little bit of math that we try to incorporate. Once we understand that more, we get into a hybrid system where we're starting to tweak the system a little bit better, trying to get synthetic portions and seeing if we can mimic biology a little bit more. And that's a little bit less biology, more chemistry along the physics area, and starting to get more into engineering. And the fourth area is where we have a complete artificial system. And that's more chemistry and material science than engineering. The program that I'm going to describe is currently in this, about this area, I would say. They're still trying to understand the mechanisms associated with this, these systems, but yet they can still tweak them, but they aren't quite there to make it a complete artificial system. Challenges for natural systems research is to explain the genetic regulatory mechanism associated with the systems that they're looking at. Another challenge is to understand the kinetics that are associated with the systems. On the payoffs, we're trying to increase the amount of energy associated with each one of the ones that they're looking at. The artificial systems, the real challenge is trying to make it as good as the natural systems are. The payoffs for that would be developing higher energy densities for enzymatic fuel cells. And another challenge would be to integrate and assemble nanoscale inorganic biomaterials. An overall synopsis of the mechanisms associated with everything I'm going to be talking about is these are the areas that we're talking. The objective for each one of these researchers is to increase the amount of lipids that they can produce, the amount of sugars that they can produce, or the amount of hydrogen that they can produce. That's what their goal was. They just want to keep trying to increase by stimulating. What they did was they looked throughout everywhere, usually in cyanobacteria and algae, to find different strains that they could characterize to see whether they had the capabilities to make the fuel source that they wanted. They looked at the genes. They connected the genes to the process that they're looking at. They developed a model, and they determined whether it increased the hydrogen, increased the amount of triglycerides, or increased cellulose. So this is the mechanism in general that I'm referring to when I talk about some of the portions that I'm coming up to talk about. So biosolar hydrogen is the first area to talk about. And one of the interesting things that happened in this field is two researchers, a guy named Don Bryant and Shui Zhang, found an error in the Cyanobacterium synococcus tricarboxylic acid cycle, the TCA cycle. They found that, or the TCA cycle in that bacterium was taught to be missing a step, a process. And these guys found that, no, that step and process wasn't missing. It is actually there, and they proved it, they found it, they published it in Science. Now they got to go back and rewrite all the books, because for 44 years it had been taught incorrectly. So that was a pretty cool finding. Biosolar hydrogen, 8 labs, including AFRL and DOE. Recently, I believe, Dr. Maybury, you can correct me if I'm wrong, but recently I think there was just a demonstration of a hydrogen, a demonstration of a fleet of vehicles in Hawaii that was just funded by AFRL, ONR, and then Army, right? And it was just run in Hawaii, and it proved successful. So they're able to function strictly on hydrogen. The objective, again, is to increase the amount of hydrogen that they could produce. In order to do that, they had to develop some equipment that would be able to measure high throughput. They looked at bioprospecting for all strains and species that they could figure out which ones produced more hydrogen than others. They looked at the systems biology and pathways, and looked at genetic engineering of each one of those pathways. The objective ultimately is to develop a fuel supply and one that's affordable. I have 2 slides on this researcher. His name's Dismukes. In this one, he's describing 4 results that he had, and they found that in this structure, cyanothese is what it's called. Found that cyanothese had 2 enzymes that could produce could turn on the hydrogen pump. It could turn it on and off. And they found that by being able to turn it on, or they found a way to turn it on by increasing or by introducing a chemical called flavone. When they did that, they were able to turn the process on, increase the production of hydrogen twofold compared to the control group. When they stressed the cell with nitrate, they showed that stressing it increased or caused the glycolytic pathway to be switched from the glycolytic pathway to the oxidative pentose phosphate pathway. Being able to do that will allow them to focus the energy here to increase the amount of hydrogen that's produced. One of the other areas that they looked at was they noticed there was in the glycolytic fermentation process, they noticed that in the upstream portion of the process there was an increase in carbon compared to the fermentation process at the base. Their objective now is to try to decrease that constriction, as they call it, right there, to be able to genetically engineer that so the carbon can flow through quicker, increasing the amount of hydrogen production capabilities. What they found in this was that NAD was able to increase the hydrogenase, and that was the first time that they understood that at the time. So they found that mechanism. Dismukes did another project where he was looking at each one of these structures can produce hydrogen in the dark, they're what's called the dark cycle, following anaerobisis. And cyanothese on the left side can produce the hydrogen in the light as well. So what the question was, could they add these two together when this one was in the glycolytic pathway, take the lactate, have it be added to cyanothese, and would it produce more hydrogen than just either of them separately? By doing that, they showed that it increased the amount of hydrogen that it produces by twofold. So the next step is, how many of those can they add together? How much can they compound and make it work? Algal oil is the next area that I want to talk about. It's the largest portion of the I'm sorry, back up one. In hydrogen, when I asked they have a murine in this area, they produced about 100 papers over the life of their murine, which was significant for that area. Algal oil is the largest area of this portfolio. 10 labs, including DOE and USAFA. Again, the object is to produce as much oil as possible. They did that through bioprospecting. They screened 1,200 algae strains for oil and identified yield and found out that 50 candidate strains for future studies that were significant in yielding enough oil to be worthwhile looking at. They studied the omics, as you'll see in the next slide, where they were trying to map the lipid pathways. They they increased algal yield by stressing the cell with nitrate also, again, to produce a stable fuel supply and amount. The omics. In this case, this person was studying the RNA of the cells, and in the proteomics, Sayer was the one that found the 50 strains of algae that produced a significant amount of oil to be worthwhile in studying. And metabolomics is the fingerprint of the cell. It leaves a unique capability. All of these communicated with each other. It's a 7-lab collaboration. They passed the information on to the bioinformaticist, created a computational biology output, and all the information is down on the web for everybody that works in this area that can use the same and start out and don't have to reproduce the information. It's all there for anybody to find and use as they want. Sayer had an interesting project where the hypothesis was that if they reduce the antenna size, the antenna is the pigmented molecule that is responsible for light in the algae plant and the algae. And if they were to, because the antenna is large and is therefore absorbing light. At full sunlight, 75% of the energy is given off as fluorescence and heat. If they decrease that antenna, would that 75% of that energy be focused inward towards the mechanism and increase the metabolic output? And the way they decrease the antenna is by increasing chlorophyll B. And that chlorophyll B decreased the antenna size, and they showed that it was increase in the 30% increase in the photosynthetic pathways, which ultimately resulted in an increase in oil production. Enzymatic fuel cells. There's 7 labs associated with this one, including AFRL. In enzymatic fuel cells, they take chemicals, they produce electrical energy from that chemistry to produce power sources, and the objective is to increase the energy as much as possible. They look at the enzymatic pathways, they try to protein engineering to increase those enzymatic pathways, they try to establish the mechanisms of electron transfer, and they succeeded in each one of those. The accomplishment, well, they developed the multi-enzyme cascades, they engineered enzymes to self-assemble in conducting hydrogels, and they determined the oxygen binding site in multi-copper oxidases. Currently, the amount of energy they can get out of enzymatic fuel cells, I was told, was just around the milliwatt level. And they can potentially run a micro UAV on these enzymatic fuel cells. The goal is to run a standalone sensor of some type in the near future. And that's at the wattage level. This is a mannequin of a person looking at a watch, in case you can't see that. One of the reasons I want to show that is because the researcher in Florida for AFRL I had talked to him, and he had said that they have what's called Wattman filter paper. They soaked it in Gatorade, and they were able to run an atomic clock for 24 hours just on the Wattman filter paper soaked in Gatorade. I thought that was pretty cool advance. So I don't know if that's 10 milliwatts or what it is, but it got some energy out of it. One of the things that needs to be done in this area is to allow the enzyme to stay on the biocathode, the biocatalyst, for longer periods of time. When doing that, the amount of energy goes up, the longer they can keep that enzyme on that catalyst. In developing the catalyst, they had to find structures that did not decrease the amount of energy utilization. In this case, this person was taking 2D-glassy carbon and coating it with polymethylene green, tested that to see if any change in the amount of energy took place. Also took 3D reticulated vitreous carbon, coated it with polymethylene green, found no change. So we took the 3D reticulated vitreous carbon as the backbone for the biocatalyst, coated it with multi-walled carbon nanotubes, also coated it with chitosan, and developed a device. This was used for the anode. The laboratory in Florida, Greg Clinton Johnson, worked on the cathode portion so that anybody working in enzymatic biofuel cells in this team could have the same type of measuring device that they had. And it showed that by taking this chitosan multi-walled carbon nanotube, it increased the amount of surface area, so the the enzyme stayed on that system longer and showed an increase in the amount of energy output from that. This team was incredibly, the Antennae team, which was from University of New Mexico, was an incredibly productive team. They had tremendous number of graduate students, tremendous number of students that worked with them. They all enjoyed what they were doing. It's extremely, what I could figure out and looking back at all the information was that they all enjoyed what they were doing. They all enjoyed working together. They started in 2006. The publications up to 2006 number about 53 or so if you're going to add them up roughly. Once they started, if you take this and continue on with that extrapolation, you can see that they were significant in the amount of publications that they put out. They did a really good job. The team is still together even though they're not getting paid anymore for it. They just like working together. They just know that they all work together real well. 99 publications, book chapters and patents. Very prolific. Did a great job. Johnson and Pachter from AFRL worked with Antennae. What they did was the same thing as what Antennae was working on, except they looked at it from a different standpoint. They took multi-copper oxidase to try to do the same thing and increase surface area. They coated it with pyrene butanoic acid succinylamide ester, or PBSE. And that's what they call a tethering reagent. And the result was that the bioelectrodes provided exceptional direct electron transport, which was significant to what they needed to do. That was a significant finding. They published in ChemCom. They were on the cover, and they're still getting a tremendous amount of publications and publicity out of this. Microbial fuel cells is the next area. Difference between microbial fuel cells and enzymatic fuel cells mostly is that the amount of energy they put out. Besides, they're done with microbes and not enzymes. Let's see. The objective for this was to understand the mechanisms involved in microbial current and to utilize the multi-scale modeling and to exploit the understanding and optimize microbes for microbial fuel cells. Benefit to be able to use microbes anywhere, anytime, for any type of fuel cell, wherever they happen to be in the world. ONR is one of the players for this in this program. They were looking at a manosensitive hemagglutinin in extracellular electron transfer. What they did was they studied this structure. They looked at the the prevailing information was that the flagellum apparently had elicited energy. They did not find that, but these manosensitive hemagglutinin pily structures right through here that you can barely see, they just look like little hairs. They measured the current and found that the current was actually in that area, and the current was significant enough to be able to try to artificially stimulate and artificially mimic somehow to where they can get the energy out of that to use it for an energy finding. So, last area that I want to talk about is the photoelectromagnetic biostimulation. The laboratory, before Walt left, he had a symposium down in San Antonio looking at this area, and they collected a group of people to find out, is this a worthwhile area to study? They know that low-level energy administered to the system does cause some changes to it. And so the laboratory has looked at some of those low-level experiences, such as direct current, such as terahertz radiation, and looking at the stimulation to the system in general to see what happens. In this case, what Dr. McKinley was doing was monitoring flight patterns on a screen, watching over a long period of time to see how people functioned. When they noticed that baseline levels were decreasing and the function started to decrease, they administered a 1 milliamp current to them and noticed that the levels came back to normal and stayed that way for a while. When I asked what's going on, even though they measured it with fMRI, the answer was, well, we're not real sure. So I think one of the things to look at is to get to the basic mechanisms, to find out what's going on, how are they doing this, what are the changes, what are the genetic processes that are taking place, what are the metabolic pathways that are taking place, how is the blood flow going, just exactly what is going on. A group down in San Antonio looking at terahertz radiation. They found that terahertz radiation can alter lipid membranes and they modulate neuronal activities. They found that they can basically blow holes through the membranes and make them big enough as they need to administer something to the interior of the cell should they want to. Once they let go of the radiation, the cell was fine, nothing happened, no residual decrement to the cell. Next thing they're trying to do is to see if they can affect the proteins or this, in the cytoplasm of the cell. Can they cause the proteins to unfold? Can they cause a change in them that they need to? Can they stimulate the DNA in some way to get the response that they want? There's plenty of questions to be asked. They're all ready for that. One of the areas that looks at this type of function is something called optogenetics, and I think that may be a way that we want to go in the future for this. Something that Walt set up before he ever funded a project was, the questions that he asked were, is the research of high quality and relevant to the Air Force? Is it unique? Does it complement what the Air Force needs? Does it leverage other agencies? Was there a team that's available to create a critical mass? He was big in developing teams. In algae, what he did, he made sure that what he was doing was not duplicated by anybody. In DOE and DARPA, they do research that is basically application funded. 6-1 work isn't. NSF funds mostly grants of individual or small sizes. EPA is interested in regulation strictly. USDA is interested in farming aquaculture. What he did in AFOSR was he developed the team and looked at systems biology, the whole process as a whole, whereas others looked at specific individual pieces of it. Nobody else was doing that. As such, he was part of an interagency working group. They all talked, they all developed what they were going to do, and they were working on the whole thing, the whole individual projects together. In biosolar hydrogen, same thing. DOE and NSF fund mostly individual grants. AFOSR's niche, again, systems biology. That isn't being done in any agency. For biofuel cells, ONR's done some work in that area. They do a great job. They showed a microbial process where they used microbes from the marine sediment. They were able to run a whole entire system with it. It's a standalone system, and it works very well. AFOSR funds enzymatic and microbial fuel cells for solid substrate and terrestrial environments that we can use anywhere throughout the world, trying to increase the power density. So artificial photosynthesis is an area that we'll maintain work in. Our big player is a guy named Dan Nocera, who's developed the artificial leaf. In 2011, Time Magazine developed or had mentioned his technology as the technology of the year for the artificial leaf. I think he came to AFOSR and gave a talk, didn't he? AFOSR and ONR. So we got a long way to go. That's what Walt had set up. Again, he did a great job. If it doesn't seem like it, it's because I couldn't present it as well as he probably could. I only had 2 months. I've only been here for 2 months. I felt like I've been studying for comps for those 2 months. I hope I did okay in giving you the background. Where we're going in the future is back to physiology. I don't know the answer to the question about chronobiology, but that remains to be seen. Any questions?
Okay. Yes.
Great job for only 2 months.
Thanks, sir.
Question related to biosolar hydrogen. You're familiar with the sunshine to petrol work at Sandia, and occurring about 5 or 6 places around the world now. One of the limitations of that work, if you will, exciting though it may be, is it's about a 15% efficient process, and also it relies upon some metals, which in various kinds, depending on the particular experimental activity, which we only can recover about maybe 25–50% of. So it's not economically viable. I kind of ask a similar question to this area in terms of the biosolar hydrogen work. You know, give us a sense. Maybe it's too early, but I'd be interested in a sort of systems view of its efficiency, of its cost effectiveness.
I'm not sure that I can answer that. I can't. I'm sorry. I can get an answer. I'll look at it and get back with you, but I can't answer right now. Thanks for the question.
Okay, any other questions?
There's been recent speculation that perhaps photosynthesis is actually a quantum effect, that you're seeing entanglement at the very beginning that moves the energy down to the leaf in this context. Is there any work of this type being supported within the program that you're aware of?
You know, I'd have to ask Dan Nocera that question. I don't know of any. We aren't funding anything right now in that, unless Tatiana, who also has a portion of this program, she was talking about some quantum work, but I don't know if. I guess she's not.
Actually, I don't know of any in this program on that, although DARPA actually has a program that they were looking at quantum effects in biology. And Tatiana, I don't know if she's here, but she was the agent of it.
And she was funding that work. So.
Any other questions? Yeah.
So you had this intriguing comment about applying optogenetics for terahertz, and I'm trying to think.
Sorry, that should have been optogenetics for the biological area of where we're going.
Oh, okay.
Thank you.
Any other questions? Now's your time. Okay, let's thank our speaker.